In the last session, we have seen some introductory part on a human excretory system. Now in this session, we will see a detailed structure of the human kidney. So that's the longitudinal section of kidney, section of the kidney. So here, to take the longitudinal section, we need to cut the kidney, okay, from mid anterior to the mid posterior. So in this way, so this is what I call the plane of division. And when we cut this kidney from mid anterior to the posterior, mid posterior, then we will get the longitudinal section of the kidney. So if you take the plane in this way, then it becomes the transverse section. So this is a TS, transverse section. Here we are not studying about the transverse section. We will see about the LS of the kidney, right? So now, okay, so when we see the kidney, on one side it is convex surface, and the opposite we have is a concave side. Now this concave side where you find a notch is what we call it the hilum. So there is a you can see depression, and this depression is called the hilum, and this is the point where the renal artery enters into the kidney and the renal vein comes out of the kidney. Okay, and that's what I'll show here. So this is the renal artery, and the renal vein also you'll see. So this is the renal artery and the renal vein. Of course, the renal artery is a branch from the aorta. So you can see here, okay. So this is the part what we call the renal artery. So this renal artery, after entering into the kidney, it will further divide uh, to form arterioles, which we will see it later. And this is the renal vein. Okay, so hilum is the location where these arteries and veins enter and okay, come out. And here also you will see one more structure that comes out of this location is the renal pelvis. So this is the renal pelvis, and the renal pelvis continues as ureter. It continues as ureter. Now, when we see the kidney in the longitudinal section, it shows two major regions. So those two major regions are, okay, the cortex, and the inner region is the medulla. So this outer region is called cortex, and the inner region is called medulla, right? And in the medulla region we find inverted pyramids. So we'll see here inverted pyramids. Okay, what these are, in a moment I tell you what actually these are. Okay, So these are renal pyramids which are located in the medulla region of the kidney, and there are around, okay, so these are called the renal pyramids, and their number is around 8 to 10. Okay, so this is approximately you will find these many number of renal pyramids. And actually these renal pyramids are the collections of the structural and functional units of the kidneys, that is nephron. So you will find okay, these lines are all, okay, are actually the nephrons. Okay, right, and this cortex will extend between these renal pyramids. So this portion of cortex that is present between the renal pyramids is what I call it the column of Bertini. So it is named after a French anatomist whose name is E.J. Bertin. Right, right after him, they have named this portion, that is, okay, the cortex that extends in between the renal pyramids, as the column of Bertini. And the kidney on its outside, it's covered by a renal capsule, is covered by renal capsule. This renal capsule is a thin membranous sheath, and this is mainly composed of collagen and elastin are the substances that are present in this thin membranous sheath and help in protecting the inner tissues of the kidney. Right, so that is the renal capsule that covers the kidney on its outer side. Okay, right, and here these columns of Bertini mean, so everywhere, so between the renal pyramid, here is one column of Bertini. You will find between every renal pyramid one column of Bertini is present. Okay, and then I said, okay, so these are all the nephrons. Later I will magnify and okay, and I'll tell you what and the structure of all this. Now the tips of these renal pyramids will open into spaces. So those spaces will constitute, so we will see something like this. Okay, so these are called minor calyx. So you will find, okay, the minor calyces, okay, and these minor calyces will open into a right bigger space, and that we call it a major calyx. Right, so the tip of the renal pyramids will open into the minor calyx, and the minor calyces will open into the major calyx, and these major calyces will finally open into the okay, the renal pelvis. So you can see here, this is the renal pelvis. So whatever that the urine formation that takes place, that will be, okay, first passed into these minor calyces, from there into the major calyces, and from there into the renal pelvis, and then ureter, and then into the urinary bladder. So that is the direction of flow of urine once the urine is formed. Now each kidney has approximately, right, 1 million structural and functional units called nephrons. Okay, so I'll just mention here. So the number of nephrons, okay, you can take from 8 lakh to, okay, 10 lakh, right? So here 8 lakh to 10 lakh nephrons. 10 lakh, okay, the 10 lakh is a 1 million, right? So that's what I said. Simply you can take it approximately 1 million nephrons are present per one kidney. So 1 million nephrons per one kidney, right? And the number of kidneys, the functionality of these kidneys will start losing from the age 40, and for every 10 years 10% of the nephrons will lose its functioning. Okay, anyway, now we will see the structure of these nephrons, which are the structural and functional units of the kidney. So here I'm going to magnify this location and see how the nephrons are actually present in that location. So this is how I have taken the outer cortex, and this is the medulla region. Now you will find here in the cortex region, okay, the Bowman's cup, so you will see like this, okay, and then the remaining portion, it enters into the medullary region, and you will find, so actually you will find two types of nephrons here, okay, right? So this is how the nephrons will be present, okay, from the cortex and in the medullary region. So I'll draw the complete diagram of this nephron later, okay. So this will be uh, the renal pyramid, how it looks like. This is the tip of that renal pyramid, which opens into the minor calyx. Okay, so here this is the Bowman's cup, and we see here the artery which enters into the kidney will divide to form arterioles, and those they enter into here, okay, and then again they form, okay, in this way. Of course here I just simply drawn it. Actually it forms a network of the capillaries in the Bowman's cup, which we call it glomerulus. So after that it comes out as a efferent arteriole, and that efferent arteriole will form a right, a network of blood vessels on this PCT, okay, and loop of Henle, and even on DCT. Now let's see here, we have of course, so finally you will find this is the loop of Henle, and the DCT, and then it opens into the collecting ducts. So these collecting ducts will collect the urine from different nephrons, so which finally opens at the tip of this renal pyramid. So this is the renal pyramid, okay, right? Now these nephrons we actually classified into two types. So one we call, the nephrons are of two types. One, they are called cortical nephrons, and another one called juxtamedullary nephrons. Now you can see in the case of, this is a cortical nephron. In the case of the cortical nephron, you will find the renal corpuscle little away from the cortex region. So here we know this is the cortex region below this line, okay, right? And in case of juxtamedullary nephrons, you will find the renal corpuscle is lying very close to the medullary region. So that is the reason this nephron is what we call the juxtamedullary nephron, whereas this one is called the cortical nephron. And the one more difference between the cortical nephrons and juxtamedullary nephrons is that in case of these juxtamedullary nephrons, the loop of Henle will be very long, okay, so it will be very long. So you can see here it's much longer than what we see in case of the cortical nephrons. So the renal pyramid, it mostly will have this Henle's loop. So here also the Henle's loop. You can see in case of juxtamedullary nephron, the loop of Henle is very long when compared to the cortical nephrons. Now, out of this nephron, what parts of the nephron they lie in the cortex and what parts will lie in the medullary region? So here the renal corpuscle, right, and then the PCT, that is the proximal convoluted tubule, then DCT, the distal convoluted tubule, and a part of the collecting duct, part of collecting duct. So these parts, whatever I mentioned here, the renal corpuscle, PCT, DCT, and the part of collecting duct, these are located in cortex region, located in cortex region. And coming to, okay, the renal pyramid or into the medullary region. So in the medullary region you will find the Henle's loop and the major portion of the collecting duct. Okay, so in medullary region, what parts of the nephron are present? So we see here the Henle's loop and okay, the major portion of collecting duct. So the major, okay, portion of the collecting duct and the Henle's loop is located in the medullary region, right? So out of the nephron, so we have seen here what parts of the nephron are present in the cortex and what parts, okay, of the nephron are located in the medullary region. So you will find, okay, so many collecting ducts collecting the DCTs from different nephrons, and they finally open at the tip of this renal pyramids. And of course what they collect is the urine, and that gets collected in the minor calyx, that passes into the major calyx and then into the renal pelvis and into the ureter, and finally gets collected in the urinary bladder. Okay, so now we will see this structure of the nephron in detail. The next one, structure of nephron. Right, so this is how we see the structure of the nephron, and here the nephron has mainly two parts. right? One we call it the glomerulus, and another one called the renal tubule, right? So this glomerulus is a network of capillaries formed by the afferent arteriole, which we'll see, okay? Whereas this renal tubule has the Bowman's, okay, cup, and then it has the PCT, so it's called proximal convoluted tubule. So in short, it is PCT, and then okay, the Henle's loop, and then we have is the distal convoluted tubule. So in short, we call it the DCT. Okay, so the proximal convoluted tubule, Henle's loop, distal convoluted tubule, and even the Bowman's cup or the Bowman's capsule are the parts of the renal tubule. Okay, so this portion is the Bowman's capsule, and this is the PCT, the proximal, okay, convoluted tubule, right? And this portion is the Henle's loop, and this portion is the DCT, the distal convoluted tubule, and this is the collecting duct. It's the collecting duct. So here I told you that the renal artery, after entering into the kidney, it will divide and result in the formation of right, many arterioles, and one such arteriole enters into the Bowman's cup, okay, and here it will divide to form a lot of capillaries, and then okay, so it comes out as the efferent arteriole. So this is the afferent arteriole, so it's the afferent arteriole, whereas this is the efferent arteriole. And this network of capillaries that formed by the afferent arteriole is what I call it the glomerulus. Okay, so now here the glomerulus and the Bowman's capsule together, we call it a renal corpuscle, is called renal corpuscle. Also it is called Malpighian body. It's called renal corpuscle or Malpighian body. Now, out of the different parts of the nephron, that is Bowman's capsule, the PCT, Henle's loop, and distal convoluted tubule, the longest section of the nephron will be this, okay, the PCT. So PCT is the longest section, right? So the PCT is the longest section. Now when you see the afferent arteriole and efferent arteriole, I have drawn, okay, with the different diameters. I have shown here that the afferent arteriole has a bigger diameter when compared with the efferent arteriole. So this is necessary to create pressure enough for the filtration of blood in the glomerulus, that we will see, okay, when I discuss the formation of urine, where we will talk about ultra filtration in the glomerulus. Okay, so the Bowman's capsule and the glomerulus together, it's called renal corpuscle or Malpighian body. Now this efferent arteriole, okay, will form a network on this PCT, the Henle's loop, and even on the DCT. Right here we can see the efferent arteriole will form a network of blood vessels on the PCT, the Henle's loop, and even on the DCT. So this we call as a peritubular, okay, network, right? I mean peritubular capillaries. So these are peritubular capillaries. Now, one blood vessel of this peritubular network runs parallel to this Henle's loop and forms a U-shaped structure, and that is called the vasa recta. Okay, so the vasa recta is a part of this peritubular capillaries. The vasa recta, this is reduced or completely absent in case of cortical nephrons. I'll give you the differences between the cortical nephrons and even the juxtamedullary nephrons. After that, we will detailedly see the structure of the nephron, even this part, okay, the Bowman's cup inside which the glomerulus is present. So, okay, what are all the layers and how it is organized and everything we will see in detail. So before that, we will see the differences, okay, small differences between cortical nephrons and the juxtamedullary nephrons. So right, so differences between cortical and juxtamedullary nephrons. So cortical and juxtamedullary. Right. So here these cortical nephrons, they constitute around 85% of total nephrons. The point here, in the old textbook they have given these values, but in the new textbook they have eliminated these values, even though I'm just giving it for information's sake, right? Whereas the juxtamedullary nephrons will constitute the 15% of total nephrons, right? So again, I'm telling that in the old textbook they removed, in the old textbook they have given these values. In the new textbook they have eliminated these values completely. Then, okay, so in the case of cortical nephrons, their glomeruli, which is a plural of glomerulus, their glomeruli are found in the outer cortex. Okay, found in outer cortex. You have to keep in mind both the juxtamedullary and the cortical nephrons, their glomeruli are found in the cortex region only. So this will be a little towards the outer cortex, whereas the glomeruli in this case is present close to the medullary region. Okay. So their glomeruli located close to the medulla, right? Then In the case of these cortical nephrons, vasa recta, which is a U-shaped blood vessel that is present around the Henle's lobe, is reduced or absent. Whereas in the case of juxtamedullary nephrons, right, you will find the vasa recta in a highly developed condition. So here the vasa recta is highly developed, highly developed. Loop of Henle is short and extends, okay, only to certain distance into the medullary region. So loop of Henle is short and extends only to a short distance into the medullary region. Whereas in this case, the loop of Henle is long and extends deep into the medullary region. So these are the differences between the cortical and the juxtamedullary nephrons. Now we will see the nephron a little more in detail. We'll see the part by part of the nephron. So first we will see about this Bowman's capsule. So this is how the Bowman's capsule looks like. So here this is the afferent arteriole and this is the efferent arteriole, and this Bowman's capsule is a double walled right cup-like structure. So you can see it has a cup-like structure into which the afferent arteriole enters and further breaks down to form a network of capillaries. So here I have drawn in a simpler manner. There will be a complex network of capillaries which we call it a glomerulus. And as I told, the Bowman's capsule is a double walled cup-like structure. It has two layers. So this is outer the parietal, okay, epithelium, or the parietal layer, of course, that is composed of parietal epithelium, and there will be an inner layer, and this is called the visceral layer, composed of this visceral epithelium, okay. And here in close contact, okay, with the afferent and efferent arteriole, you find the distal convoluted tubule, and this is the lumen of DCT. So that is distal convoluted tubule. The space between this parietal and visceral epithelium, so here this we call it the capsular space, okay. So into which actually the primary urine formation is released here, or the filtrate enters into this. So here is the location the filtration takes place, and this will be the thickness where the filtration actually takes place. And once the filtration is completed, the substances will enter into this capsular space, and that run into this PCT, that is proximal convoluted tubule. So here there will be three layers through which actually the filtration takes place, and that's what we call the glomerular capillary blood pressure, okay. So the glomerular capillary blood pressure causes the filtration. Okay, and here the filtration will take place through three layers. So here you find total three layers. So we will see those three layers, how they actually are present. Okay, so you can see here the afferent arteriole. So it has endothelium layer. So this is the endothelium layer. Okay, so endothelium of glomerular capillaries. So it's an endothelium of glomerular capillaries. Okay, and this you can see is the epithelium, that is the visceral epithelium of the Bowman's capsule. So right, the endothelium of glomerular capillaries and also the epithelium of Bowman's capsule. So this is epithelium of Bowman's capsule. Okay, and between these two you find a basement membrane. So you will find a basement membrane. So basement membrane. So this basement membrane is present between the epithelium of Bowman's capsule. So this is the epithelium of the Bowman's capsule, and this will be the endothelium of the glomerular capillaries. Okay, here I'll just magnify this and show you that how the three layers are present. Okay, so this is the capillaries. We know the capillaries will have endothelium and a basement membrane. Okay, right, and this is the epithelium of Bowman's capsule. Right, so this is epithelium of Bowman's capsule, and this is the capillary. This is the lumen of the capillary where the blood flows. So blood flowing, okay, in a direction, and this is the endothelium of glomerular blood vessels. Endothelium of the glomerular blood vessels, and between these two layers is the presence of the basement membrane. Okay, so this is the basement membrane. So now the filtration has to take place. Okay, so through these three layers, so that is endothelium of glomerular blood vessels, then through the basement membrane, and then through the Bowman's capsule, and then the filtrate enters into the capsular space. So these are, okay, total three layers through which the filtration takes place. Now here the epithelial cells, so that is which are present in the epithelium, the epithelial cells of Bowman's capsule are actually called the podocytes. So they are called podocytes, and these podocytes will wrap around, okay, the glomerular blood vessels, and in this okay, when they are wrapping around the glomerulus, So they actually leave some minute spaces, and these minute spaces are what we call them the filtration slits or the slit ports. So here in this diagram is I did not show here, right. uh, in the next session I will show you that how these podocytes will be wrapping around the glomerular blood vessels, and how they are going to leave the spaces between them, which we call the filtration slits or slit ports, actually through which the substances will get filtered and entered into the Bowman's capsule.